Oh, wait. Yeah, I do.
It says you're speaking.
Hi, everyone. This is Ellen. Should we go ahead and take attendance? I think we're about a minute past.
Yeah, let's move ahead. Thank you.
Okay, great. Who do we have from Broad Proteomics?
This is Adam Officer.
Okay. Who do we have from Broad Transcriptomics? Okay. Who do we have from HMS?
Hi, Carolyn's here with Jeremy Mullis, Sean Erickson, and Dave Roble.
Hi, everyone. Who do we have from Detox Mount Sinai?
Hi, this is Eric Sobey.
Okay. Who do we have from the DCIC?
Hi, here in Miami, we have Michele, Vaz, and Stefan. Anybody else on the phone?
Yeah,
This
Daniel's
is
here too. I'm at my desk.
Oh, Danny is there as well. Yes, yes, of course, we need him.
Kathleen from Mount Sinai and Avizan
Also, Ali.
and Ali.
Okay. Okay.
Kai Young from the EDSR, the DCIC.
Okay. Great. Who do we have from OHSU?
Jim Clark,
Laura Heiser.
Damir Sura.
Estelle Nebula.
Joe Gray.
Okay. And who do we have from Neuralink?
Kelly and Maria from UCSF Gladstone.
And Steve Finkbeiner from Gladstone.
Okay. Thank you, everyone.
All right, thanks everybody for joining. Oh, we forgot from NIH. Does anybody, SR Albert? I'm not sure if RJ is there, but just, I don't know if anybody else is there that you want to mention with you. Okay, all right, so thanks everybody for joining. So the first agenda item as usual, approving the minutes from June. Alan sent them out and they're also on the site here. So are there any disagreements? All right, so if not, then we approve the minutes. And then what we want to do today is, so we have a presentation later from OHSU. I mistyped this in the agenda, apologies for that. But first we're going to go quickly over the metadata standards. So we have, last time we discussed how do we solve the problem that we have reagents that are small molecules, but we don't want to kind of put them into the small molecule collection because they are not perturbagens, and because of that they get a little bit of a different treatment. They're just operationally differently handled. in the DSGCs than other perturbagens for molecules are. And in order to address that, we discussed last time we're going to include a new category. And Danny, do you want to quickly go over this here? I can link you to the proposal here. I show it on the screen. And essentially, this is the same thing that was proposed last time. We don't need to repeat that, I think. It's basically just describing the problem that we have some dyes, right, and what we want to do. And we discussed this last time. And now, basically, here's just a specific thing that we would like to do in that link document, fairly straightforward. But Danny, do you want to mention it really quick, or do you want me to mention to explain it?
I can go over it really quickly.
Okay, can everybody hear Danny?
No,
No, you can't. How about now? Is that better?
not really.
Hang on. How about now?
Barely.
Okay.
So, let me, why don't I just go over it? I think it's really quite straightforward. I mean, I probably can't do this quite as articulate as Danny, but I will just try. So essentially, basically, we have a fairly simple proposal here. What we're going to do is, and again, this is because perturbagens are, I think, operationally somewhat more important than other auxiliary reagents like dyes or staining reagents. Not suggesting that they're not important, but it's different. So what we want to do is basically introduce a class unclassified perturbagen, and we would move perturbagens that are not small molecules and that are not otherwise classified, for example, not antibodies or microenvironments or genetic perturbations, into unclassified perturbagen, and then use this category that we have for other reagent, use that for those auxiliary reagents, right? So for right now, we have, for example, another reagent, we have lipopolysaccharide, for example, as a perturbagen, and a couple of other perturbagens in there. And in order to maintain some kind of clarity between the categorization by molecular type and its role, we decided to, in the last meeting we discussed that we should kind of keep it relatively straightforward. So we are thinking we will describe the small molecules as small molecule perturbagens. So only put in there those small molecules that are actually perturbagens that are used in assays for perturbation, and then we have other categorizations, of course, in the metadata, and then add this unclassified perturbagen category in here, and then basically move some of those that are currently in the category that's called other reagent over to unclassified perturbagen. We would then have to change the prefix to UP, I guess, and then reserve this other reagent for these auxiliary reagents, right? So, yeah, that's what we are thinking so far. Does anybody have comments or kind of concerns about that? I mean, there's this one drawback that we're gonna have to change, like, I guess, eight IDs by changing the prefix. I think so. It's probably good to do it because we maintain this clarity about what's what, and we don't mix up auxiliary agents, and other perturbations and so on in just one big group that's called other. So, do you guys have any comments or concerns about that? And
It sounds great. I think that's wonderful. This is Carolyn.
Right. Okay. So that's great. So if you guys think it makes sense, then obviously we will just give you a mapping to those if any IDs change, and this will of course be minimized, right? So and then now the unclassified reagents, Vas will just show this in a minute, and then for the new now other reagents, we just basically propose a couple of like very basic fields below that you see here that I don't need to repeat now, right? So if that makes sense, then if I move back to the agenda, maybe not the one from last time, but the one from today, then we thought we should just take the opportunity to also just, if we change the reagent metadata anyway, we have a few little changes like typos, and some little updates, corrections in the reagent metadata that was put together, and they're here in the summary. And then that summary also includes those fields for the new unclassified perturbagen. So Vas, do you want to quickly go through this here? I'm going to open this field and give you the mouse. And then you could maybe quickly go through it. But I mean, it's the main message here is basically just minor things. And we just wanted to not just make the change, but basically show it really quick and then give everybody the opportunity to just look at it.
So yeah, in this Google Sheet, I put all the metadata from previous year that we had agreed on. And I color-coded some typos that I found, and we found here, for example, list was lowercase, but we start our definitions and comments with uppercase, so to be more consistent. And I think the idea would be if you guys can browse through, maybe I think in a week, to see if there may be any other typos that were found. The most important ones I've highlighted with the red here. I have the column that says fields with changes. So you don't need to even run through everything. You just focus on the ones that had a red column. And lastly, this is the new classification. So like we said, we're going to rename the previous other reagents into unclassified perturbagens. So whatever was there, we're just changing the prefix from OR to UP. And then we're going to create the new category that stains, buffers, and all those auxiliary secondary reagents are going to be. And these are going to be the new fields. So secondary antibodies or stains and buffers actually are going to be here. They're reagents that are not, they're not perturbagens. So I don't know if maybe if you find any other errors, maybe you can comment. This is
just to be transparent, right? So the plan would be to essentially just make another release and have those little errors there fixed, right? And then I think you're adding this comment there. Right?
So yeah, we wanted to be consistent again with all the classes and all the categories. So this is just a generic field that's going to be in every category. That's called. There's going to be comments that haven't. So it's a placeholder for anything else that we haven't included.
It's a batch field, right, for everybody.
Yes, and it's a batch field.
So essentially, it's just to add some additional information that could be relevant, right, to
the reagent, but we haven't
that's free text, right?
Yes.
Yeah, okay.
And I think that's about it. Minor changes mostly.
Yeah. And then, I mean, the important part, I guess, is here this unclassified perturbagen, right? So this is basically similar to small molecules, right? For perturbation, what information we want to have, right? So can we say, I mean, we can look at it now, but I don't think we need to go through those in detail. It's really fairly straightforward. I think what we should do is maybe if every group, and you could put it in the minutes, could look at this in the next maybe 2 weeks or so, give us any input or feedback, and then we will just release it, So I preferably in a week if you guys can do it, but we wait 2 weeks. And then we will just go ahead and release them. I think it's probably okay. I mean, I don't think it's necessary to look at this in detail, but of course everybody should have the opportunity to look at this and give input.
This is Carolyn. I did have a question about the antibodies. They're not only antibody reagents, right? They're antibodies. Antibody perturbagens. They're antibody reagents.
That's right. So they are a little bit special, right? They have.
Right. Okay. Just wanted to confirm, right? And actually, I think we keep our secondary antibodies in there. Do we have to put secondary antibodies in other reagents, or we could put our secondary antibodies with our antibodies if we wanted to, right?
I was thinking so. Yeah. So we should probably be consistent. So what? I mean, can we just put them?
We wrote the antibody standards there to take into account that you could specify primary or secondary.
Exactly.
Right?
I remember that too.
Yeah, so we should keep all the antibodies there and not put the secondary antibodies in the other reagents.
So yeah, there is a field here that specifies if the antibody is going to be secondary or not.
Right. Yeah. Okay, good.
Yeah, so let's put them in there. You know, I think it would be good if we are consistent in the. You can put this in the Vas. Can we put this in the description of other reagents? Maybe, you know, if we describe the categories, you know, like.
Oh, okay, yeah.
Let's make it a little. You know what we're gonna do is we're gonna write a little. We probably have that somewhere already. A little description of all the categories.
Yeah, I think that's a good idea.
You know, and then we basically say what goes into each, and then we share that as well in the next. You know, let's share in the next couple of days. You know, and then we can review that together. Okay?
Yeah, that's a good idea.
So we can do that, and then so just everything is clear because it's not getting easier, right? So we have now small molecule perturbagens, right? And it's a little bit it's not completely trivial, self-explanatory. So let's do that. Okay. So any other comments? We don't need to drag it out if you don't have to. Okay. All right. So I think what you're gonna put again, I summarize really quick. So DCIC will generate a brief description for all of those. And then everybody can review them in the next 2 weeks. And then we're going to release the next release. And then what we're going to do is map over the previous other reagent perturbagens into unclassified perturbagen, and then move those, and then those reagents that don't fit any other category and are not perturbagens go into other reagents. Yeah. Okay. All right. So okay, that brings us probably to the more interesting part, the presentation from OHSU. I did actually correct this here. Let me just refresh. Okay. All right. So so I think it's Damir and Michael. Can you share the screen and to show the slide?
Yep.
Yeah. So
right now.
So I'll go first. And give me a second here. Okay. Right. Good. Can everybody see my screen?
Yeah, looks good.
Okay, good. So the idea was to give an overview of our recent changes and work that we've done in image data analytics and management. And since the management kind of goes over it all, I'll start with the management component, and then Michelle will take over and discuss the analytics, which is probably the more interesting part. So the image data management that we've, the structure that we've built is specifically for our map links project. But as you'll probably see when we get through it, a lot of the components are actually very generic and could be used by anybody who does image data work. And specifically, the HMS group is using many of the same components that we use. So you'll see that a lot of it is actually kind of the same type of stuff. All right, so since what we are talking about is specifically on the MapLinks project, a very quick reminder what the project is about. So it is about systematically assessing the microenvironment components as perturbagens, and we do that by spotting high-density grids in microtiter plates of insoluble components of a microenvironment. We seed cells on top of that, then subject the cells during their growth phase to a soluble microenvironment in each well. And so what you get is sort of the effects on the cells during their time sitting on a insoluble component and a ligand, a soluble component, at the same time. So that gives you a combination of two perturbagens, and in our current array, we're doing this for about 2,500 different combinations. And our plan is to do this over a panel of about 20 cell lines. And I think we've currently hit about 6 or 7 of them. And so we're still going through them one by one. The readout that we do is multiple, but the most important one that I'm going to talk about today is the imaging readout. And so after the cells have grown, they are stained for a number of biological endpoints using immunofluorescent staining, and then the plates are imaged, and the images are subjected to our management and analytics. So that's what we're going to be talking about today. This generates a very, very large amount of image information, and so doing this in a systematic and structured way is quite important. And image data in itself, it has already many different challenges, which we've kind of talked about before, so I'll not go through this whole list. But one of the important things kind of at the end is images are pretty to look at, but actually they are data. And when you actually want to understand the data that is derived from these images through the analytics, it is at every single point throughout important to be able to go back to the images themselves. And so what we found is for our in-house use, it's really important to keep that link close to the image data itself. But actually what we've also found is that people from the outside world who want to see our analytical data will also want to be able to visit the image data that underlies it all. So it's important to have access to this data, the images themselves as well. And so what we've built on is a open source kind of community effort called OME, the Open Microscopy Environment, and then specifically its implementation of a scientific image database called Omero. Again, we've gone through this before in previous talks, so I won't belabor this any further. It works well. It's installed throughout OHSU in multiple projects. It's also used heavily at Harvard Medical School and a few other sites that people collaborate with. So I think it's a good platform to build on. So one of the things that it builds that's built into it is a capability to handle imaging metadata. Here it's maybe useful, since we just talked about metadata a bit, to go a little bit deeper into that. So the idea is that we have 2 types of metadata actually that are part of imaging. There's the image acquisition metadata, which is typically generated during the imaging process, right? It describes how the microscope was set up and how the imaging process itself took place. So that data tends to go with the images themselves. It's generated effectively at the same time as the images themselves are created. It is very critical for interpretation, but typically this is not the type of data that you use for searching for something of biological relevance. It is more the information that allows you to interpret your images. The other data, and that is more the type of data that we've talked about in the last few minutes, is the metadata that describes the experiment. And so that tends to be created not during imaging, but before already. It describes how that sample got to be where it is. And it tends to be very critical for search and selection. It is what you want to search on. In the ideal case, like was indeed described just now, is it should link to authoritative vocabularies and ontologies. So what the DCIC is doing in Stefan's group is really important, because it allows everybody who uses this data to actually say, yeah, it means this, and we all agree that it means that exact same thing. The nice thing of having an image environment that handles all of this data, like Omero, is that these 2 types of metadata can be in the same infrastructure and can be managed at the same time. And so that's exactly what we've done. Then going on to how our kind of data flow currently works. So I think you can see my mouse pointer. So here on the upper left, we have the image acquisition pipeline that loads the data ultimately into our central Omero database, where the images, the thumbnails, and the imaging metadata is all stored. Then our second pipeline is that experimental metadata pipeline that is built on our in-house ANOT LIM system that tracks all the other data, the experimental metadata, and it also goes into our Omero database. Then on the right-hand side is shown kind of components of our analytics, and Michelle will go into that much further. But the idea is that all the images, the metadata, the segmentation results, the feature measurements, all get stored in our central Omero database. And that's all good, but of course it does need to be available to the outside world as well. And so we've started working with, we've been working for a while with having ways to make the data available to the outside world. So we have the Synapse site, which I'll touch on very briefly. But recently we've started working on a project, we've made good progress on a project to make our image data available as well. Synapse was not really the site to make the image data itself available. Our publicly available Omero database, which is sort of a copy effectively of our in-house Omero database, is specifically meant for that. So that is our latest, newest thing to announce, is that we now have an imaging data open access site. So in-house, we have collected a huge amount of data. It's about 80 terabytes in-house. That is located at OHSU behind a firewall, so it's not particularly accessible. And in order to make it available to the outside world, we've deployed a second copy of Omero onto Amazon Web Services, AWS. And that is now up and running and available. And we've loaded a subset of our data, the May-June 2017 release that's our most recent release. It's about 10 terabytes worth of image data and associated metadata and feature vectors and everything, which is now all sitting on that publicly available site. So that's available on the URL omero.linksclarion.org. And it's built on a whole detailed infrastructure that I won't bore you with today, but if you're interested in setting anything up like this yourself, please come and talk to us. This is, of course, still just an Omero server, so it doesn't really give you very many friendly tools to manipulate your data. So the second part that we've built, and that's not yet up, but very soon, is a portal site, which is going to be and kind of is available on www.linksclarion.org. And that will give you all kinds of nice tools that they'll give you a very brief demo of. So that is what the world will eventually see and currently actually can see is direct access to our full image data and all the metadata directly from the Omero server on the cloud side, and also a nice user-friendly portal that gives you the same access to that same image data, of course, but with some nice visualization tools sitting in the front. So those visualization tools, there's a number of them that we've already built and that just need to be collected together and put onto the site. And when I say we, that means an effort from a number of people. So on the upper left is a set of apps that are built by Mark Dane at OHSU using the R and Shiny capabilities. In the upper right is a very nice visualization tool that was built by Avi Mayan's group at Mount Sinai that takes the image data and the associated metadata and has a very nice interactive viewer. And then a commercial tool that we've deployed on our site and will eventually be making available is a very image-centric viewer called Sagami that allows you to deal very directly with the image data itself. And mostly I'll just skip through these things very quickly, but let me see. How do I get out of here? So the Links Clarion site, as I said, the portal is still under construction, but when you click on the link through, you get immediately to our image database, and for every plate that we've imaged, which is all very well laid out on the Synapse site, you can get to any of the images that underlies the data that is on the Synapse site. And for each image in the database, which unfortunately loads a little bit slowly because I'm still loading data into it, so it's a little bit busy at the time. There is associated with each of the images a whole bunch of metadata, which is shown here in the key value pair. So that's searchable, and in tables that are downloadable and can be used for analysis. So these are all the measurement data per image that are also shown on the Synapse site. So that's the Omero in the cloud instance. Then we have our Zagami viewer, which is as I said, a very highly interactive viewer that allows you to very rapidly look at very large numbers of images and use the image views to get a feel for what the data looks like. So what I've just done is I've taken one whole plate that had 8 wells with different ligands, and I'm kind of looking at these individual images on a per ligand basis, but putting them together. And then I can say, well, I only want those that have a reasonable number of cells in each spot. So I'm sort of sub-selecting, and I can do quite a bit of interactive analytics using this. The next thing is the interactive viewer that was built by Avi Mayan's group, which takes the image data and the metadata and does very rapid and interactive PCA analysis on these data. Has also capabilities to do Tisney plots and cluster plots, and you can, by looking at groups of data together, you can get all the feature vectors of each condition, each perturbing condition, together with others that looks very similarly. So there's quite a bit of nice interactivity here. In order to not go way over my time, I'll skip the last 2 little demos. But when I put this,
actually, I think you can go ahead if you want to. There's not a particular time constraint you have here. You have enough time because we went early. We went quick.
I want to give Michelle time, so I'll stop here. But this presentation, I presume, I'll make available to put on the website, so people can go to these URLs themselves and look at more of the tools and the image data that we have available. So thank you very much. And Michelle, if you want to take over.
Thank you. Yeah, great. So let's wait for questions until Michelle is done, right?
Yeah, that's a good idea.
Okay.
So I don't know. Do I need to release, or I guess I've released. Good.
I think I kicked you out, right?
Yes, perfect.
Okay, good. So no questions? At the end. Oh, okay. I thought you were taking questions in between. All right. So I will talk specifically about the image analysis aspects, and there are a few things that repeat on the infrastructure. So I will try to go through the first couple of slides very quickly. As Amir mentioned, we are in parallel acquiring an enormous amount of images that get stored in the Omero database, as well as collecting metadata every step of the way during the experimental procedures that are happening in the lab, using the ANOT database that Elmar Boucher wrote here at OHSU. And what we'll be talking about is the part all the way on the right of this diagram, that is the analysis of the images and the extraction of the features. So in the first 2 years of MapLinks, we've scanned 309 plates in this fashion and collected millions of images and billions of features, literally. And the way that this has been collected, processed, verified, quality control, checked, and further explored is laid out in this diagram. That's sort of the simpler version of what you saw earlier. So the Nikon scanning system is producing all these images, and I'll specifically be talking about pulling those images then out of the Omero database and analyzing them with QI software. That's our in-house software, as well as the Cell Profiler software. And as you've seen before, on the right-hand side, we have a number of viewers and further analysis tools available in our infrastructure at OHSU here. And some of the reasons as to why we're doing that in this fashion is because it's optimal for links, but also it fits in with our larger collection of microscopes here at OHSU and other analysis and visualization tools that are floating around here that may or may not be related. So the bigger picture of this looks more like this, where there's a dozen different microscope systems and at least as many different visualization tools and analysis tools available. So, but specifically, we're talking about the imaging workflow where we're using Cell Profiler to do analysis of the 4-color images that are coming off the microscope. And there's a controlled and managed MapLinks box in the center here that we have set up to manage this image flow, take care of the metadata, convert things to different formats as needed, curate those features and compare them, and essentially start and stop all these various processes that are happening. The front end of this is a dashboard where we can control and visualize and start different programs up. And the end result of it are published on the Synapse data collection site eventually. So this is all way before that actually happens. And in fact, I'm talking specifically here about the phase where we're analyzing the images and extracting the features, and I'm not covering the part that Mark Dane is doing, where he actually is the recipient of these feature files that come out at the bottom of this workflow, and where he does the statistical analysis and normalization and report generation that is all happening after the actual image analysis process is done. So on this diagram, we have two different workflows. One is the Cell Profiler workflow, which is generating all our data. And on the right, we have a quality control workflow where we additionally analyze all the images and make sure that the segmentation is working correctly and the features are corresponding. And it's also a place where we can go in and experiment with alternative and new approaches to segmentation, as well as to exploring creative new features that may not be readily available in the current Cell Profiler release. So, and actually this is not happening just once, but it's happening in parallel with many runs of these Cell Profiler instances. And the same thing on the QI side, we actually run it in parallel on many, many different plates. Because as I said, we have a lot of these plates going through, and there's literally millions of images that can get processed over a week's time. So the basic idea is that we control the Cell Profiler runs from our dashboard, and in order to do so, we have a way to define the staining sets for the different experiments and to define the Cell Profiler pipelines. If you're not familiar with Cell Profiler, the Cell Profiler pipeline is the script, essentially, that is the recipe for analysis. It contains all the parameters, how to segment the cells, and it contains the specific as to what it should be measuring and how it should be exporting those data measurement files. And so we'd like to control that because we need to run a lot of these, and we have a lot of different staining sets and a lot of different experiments. So our dashboard was created so that people in the lab can easily define new staining sets and we can run a large number of these processes in a fairly automated way. We also monitor the output of Cell Profiler. So normally the results would come out of Cell Profiler as a data table, but we'd like to do the quality control on the results. So not only do we look at the output data itself, but we also look at the segmentation results from Cell Profiler, and we vectorize those results and bring those back into to our database so that we can do quality control, make sure that the nuclei in the cells are segmented properly, and we can also do post-processing on the feature table so that they adhere to our standards, and sometimes we drop a feature that is not interesting, or sometimes we may compute a feature that is a combination of other features. So that's all happening in a post-processing step. So why are we actually doing that? Well, the thing is that image analysis is not perfect, in particular segmentation, when cells get highly confluent or things are starting to overlap in three-dimensional space on the microscope. There's errors that can jump in. So if you look at cell number 37 on the segmentation side, this may have been interpreted as a single nucleus, whereas in reality it was two nuclei that were just very close together. And so you will get different data, and that's the reason why we are doing this significant effort in quality control to make sure that if there is error, it's not happening too often, and if there are too many, we can actually do multiple steps to generate additional data about that plate. Currently, most of the features that we're measuring are simple expression features, so total intensity inside of a nucleus of EDU, or total intensity or average intensity of cytokeratins in the cytoplasm. So our current drive is to look at more advanced features, to look at expression patterns and localization and co-localization more specifically. So right now we're measuring 140 features per cell that are primarily biomarker expression levels. And in order to visualize those things, we have developed a number of tools to move around in the data space and in the image space, and I'll talk specifically about the image space here. So first of all, quality control on anything that is plate level requires a map of the plates and a color-coded heat map of features that are derived on each spot. So the key thing here is that you should not be seeing particular patterns on the plate architectural level because the samples are printed randomized. So if you see a pattern, then it's probably an artifact. And the next thing you want to do is then immediately zoom into the images, of course. So this is sort of a Google Earth deep zoom viewer where you can then look at the plate layout, identify spots that may be suspect because they have too many or too few cells present, and then you can zoom in and out and try to find the image data as well as the segmentation masks that were associated with that. I'll jump on. So the other way to look at it is not on a plate architectural centric entry point, but rather looking at feature data as an entry point. So we would like to be able to have a graph of any of the features that we measure and be able to gate or just click on a data point and see which particular microenvironment spot gave rise to that particular data point. And so there are a number of tools where we can plot, and we can click on data points, we can draw these areas, and then pull in the corresponding images, and also pull in the corresponding metadata that tells us what the microenvironment molecules were that were associated with that particular NEMA experiment. And of course, that's an almost endless quest to get the best possible combination of feature plots and interactivity to do gating and interactive ways to pull in all the image data, preferably at a high resolution level, not just thumbnails, right? So you see on the top right, we have a heat map where we can do similar kind of things. And this becomes rather complicated because if you look at the mechanical background of this story, the simple plot on the user screen with all the data points can point to eventually a single cell in one image, but the one image comes out of a big file that actually has 700 images that is much larger, and that comes out of a plate, and that comes out of our plate library. So to do random scatter-gather access to any cell or any MIMA in our very large data set, which currently has 309 plates, it's quite difficult to build the backend that will allow to serve up any cell or MIMA spot on an instant notice. So what we've done is create a way to scatter, gather, and pull these things in over time as our own backend here. And then we started working on ways to do so for the rest of the world, because what you see now only works inside of the OHSU network boundaries. So the next thing was to build the Clarion website that Damir introduced. So as you see, we still have the same server and the Omero image database in our own lab here that you just looked at. That gets copied to the OHSU Advanced Computer Center and then copied to an Amazon website, and then it's accessible through the Clarion website that we have been building, hopefully with the ability to do similar things as you just saw. And the DCIC will have an entry point to Clarion so that you can just go and find, why is it doing this? So that you can just go to the front page and start exploring these microenvironment perturbations yourself. And the idea is that you can go in and select your own MIMA experiment. So you can pick your cell line, you can pick the ligands that you're interested in, and you can pick the ECMs that you're interested in. And then based on that, it will sort of pretend it's going to do all these experiments for you, but it actually is already done because we have all the data loaded up on the website. And then you can go in and look at these images just like I showed you and do similar graphing and gating functions that allow you to find a subset of the MIMAs that are of interest to you and a subset of the particular ECM ligand combinations that you might be interested in for your own experiments or in general for research purposes. So what we're looking at here are a number of screenshots of the Clarion website where you can zoom in to the images, gate on the graphs that you see below, and build subsets of the MIMA images accordingly. When you're done with that, you can actually download the images or the data and do further analysis. So this is a conceptual overview of all of that. You can pick your microenvironment experiment scope. You can link to DCIC related experiments. You can create these scatter-gather image views and the different plots. You can look at the TensorBoard cluster grammar to further explore the data. You can look at the Omero browser or the Zagami browser, and you can get access to the reports that Mark Dane is making that are available. And then in the end of all of that, you can press the download button with essentially a shopping cart of images and data that you've built up in this session. And then if it's a lot of data, it will do an out-of-session file transfer using the Globus network interface. And that's the fast version of of this story. So I'll stop here, and if there are questions or details, I can jump into more detail if you like.
Thank you. So that's really amazing work. Yeah, so questions? So I have a question where I wonder, like, so that's really cool. So you know now, so I wonder to which extent it would be easy in a way if you come from those data from somewhere else, right? So let's say you have other data integrated from some other tool, and then you want to basically get the corresponding images via an API or via just the metadata, the experimental metadata. Would that be also something that would be doable?
Yes. Shall I
Yes, in our local setup, we have the ability to import additional data and attach that to the images. on the Lynx Clarion website currently. We ran out of funding. We only had some pilot funding to actually build this.
I'm not saying that I'm not saying you don't have enough features there. You know, that's really amazing. I was just wondering, right, if you know, for example, you come from some other analyses, right? Like let's say from that is one example from this image, from this viewer, right? From RV, and then you would maybe like select some of those and would say, okay, let me get those images.
Yeah, so let me answer that one. Indeed, Stefan, you're right. The image table, the feature tables, as they are distributed through the Synapse site and through pretty much any outlet that we have, has in it also a identifier that identifies the image. And from that image identifier, you can, using a very simple URL, you just construct a URL from that identifier and prepend. You can pull up any image that we have on the Clarion site and display it as a kind of a floating window, or if you have, if you do this through R or through Python, you can just pull up the image and display it in your
oh, that's great.
Program.
So that's facilitated via Omero, right? To have the ID.
Yeah.
Okay.
Omero ID.
I get it. Yeah. So that's fantastic.
Yeah, and not only the image data is retrievable that way, but you can also ask for a JSON version of the metadata that's carried along with that image. So with that same ID, you get both the image and the JSON blurb of that metadata again.
So we should definitely take advantage of that, and I guess talk to you with some more to make sure that's also somewhat facilitated via the Lynx data portal, right? So you can basically get to the level 0 data to the images.
Yeah, definitely.
And that would be a convenient day, or at the very least, you know, one could. But we don't discuss it here. We should talk about it some more.
Yeah, definitely. Happy to discuss.
Yeah, great.
Yeah, hey, Damir, this is Steve Thinkfinder. I wondered if you could just say a little bit more about the visualization tool that sounds like Avi developed. I was sort of curious, what were the features that Avi was using to do the PCA or to do the clustering?
When we run Cell Profiler, you get like hundreds of different features on a per cell or per image basis. That tends to be a little bit too much. So what we've done in-house is sort of decided which features are kind of biologically useful to report out. And so that is a fairly small subset of about 20, 30 different features that in our opinion kind of describes the images reasonably well. So that feature vector, so on a per image basis, we have this feature vector of about 30 different descriptors with quantitative numbers. And that is what goes into the PCA analysis that Avi's group has done. And then this TensorBoard viewer displays that in a very nice interactive way and has the capability also of pulling in the kind of JPEG versions of the images. So these are not the full, many gigabyte images, but they're just JPEGs that illustrate each of those conditions. And so that is what goes into the TensorBoard.
Great, thank you very much.
I can add that there is, with TensorFlow, Google released a library, an open source library to visualize high dimensional data called the TensorBoard. So Anders, that was in my lab, he took that library and he customized it to visualize the level 4 image data. And then using the reference of the level 4 data that just has the processed features from the images, he retrieved the images through the Omero browser using the API.
Exactly, and that's that same API that we talked about just now.
Yes. I also want to say that great job today, and it looks really nice, beautiful work and beautiful website. And we would love to advertise it once it's ready, and also test run it. So if you want us to, also give you some feedback on once it's released, as from a user perspective, we can also do some QA for you. And
Perfect.
then once it's announced, we can advertise it on the Links Project website and Twitter and all that.
Absolutely, yes. We're looking forward to that.
Yeah, we're looking forward to that. Of course.
So, any other questions or comments? I mean, it's again great. So I think we should, if we can do this, link the slides to the website. I can do that if you send me the PDF version or any version, and then everybody can have access to it within the DWG.
Yes, I'll be uploading it for you.
All right, thank you. All right, so any other questions?
This is Albert. I have a question. It's sort of a metadata question. So I'm sure obviously the metadata construct that describes the images at OHSU quite well. How generalizable is it to potentially accommodate images generated from the other centers?
Yeah, that's a good question. So we have started working together with Stefan's group on defining a metadata structure for imaging data. And of course, since we're doing this together, we're trying to make sure as much as possible that as far as the image acquisition metadata, that what we are building is the same as what the DCIC is legislating, let's call it, or proposing. As far as the experimental metadata, all of our experimental metadata also adheres to the DCIC's ontologies and vocabularies. So that in itself doesn't say that it's immediately applicable, because of course our set of molecules and perturbing agents and reporting entities is going to be different from other people's, but at least ultimately it all comes down to that same base descriptor set that is held by the DCIC. And the tools that we use to annotate the images, those are all very generic tools. So those should be really straightforward to adapt to any other environment that also uses perturbing agents and reporting molecules and antibody staining protocols, et cetera, et cetera.
That makes sense. No, I mean, this come together. You pulled together a really nice effort in a relatively short amount of time, and actually the search interface looks really impressive. It's a very, very nice job.
Thank you. And yeah, I'd love to have people get their hands on it so that they can actually use it to do something useful.
All right, so, well, thank you so much. Again, great. So I think we're running out of time here. So we have 2 more minutes. It's not enough time to do the data set citation. I think what we're going to do is, again, the slides are here. The point of this is just basically it's already implemented in the Links data portal. So basically data sets can be cited, and this is possible using the collection, meaning all the data levels together, and also like the individual data levels. So we will present it next time, or we will present it maybe in one of the web page meetings and links that run on Fridays every month. I don't know exactly which Friday, but those who are on it, they will know, and probably Alan will send it out. But I don't think I want to go over time right now. So either on one of those Friday, the next one, or in the next data working meeting, we will show it. But essentially, and you can look at those and look at the Links data portal at the dataset citation is there. And of course, comment, let us know what you think. All right. So, is there any other business, anything else you want to bring up, or anything else important you want to cover in the next coming meetings? So, if not, we will send out the minutes as soon as possible, and an agenda for the next one as well soon. So can be prepared. I just want to look really quick if you look at the presentation schedule here. Really quick. Let's see.
Tony canceled our meeting, so take your time. Oh, sorry.
Let's see the next meeting that we have. So it would be on September. That's not really correct, right? Our data working group meeting is in the Bethesda meeting is in October. So we will figure out who next time it looks like. It could be DCIC presenting, but we will figure out who will be the next presenting next month in September. All right, then. Thanks, everybody. And we meet again in 1 month. Okay. Thank you. Bye.
Thanks. Bye-bye.
Thank you.
Bye-bye.
That is cool. You should look at this. Some more.
I can send you the tensor board.